Hi, everyone. Thank you for joining us to our webinar mini-series today, Single Cell Sequencing for Every Lab. This webinar mini-series includes 3 sessions. In today's webinar, part 1, we focus on current single-cell sequencing technologies. In this webinar, our expert will help you understand single-cell sequencing workflow, comparison of current technologies for single-cell sequencing, advantages of portable devices for single-cell sequencing. This interactive webinar follows by Q&A section, so you're welcome to ask any question by writing into the Q&A section. You can contact us also directly for any project-based consultation afterwards as well. The webinar will be uploaded to our YouTube channel in the following days, and this webinar is being recorded as well. So today we have Dr. Murad Shekar, so he is our today's speaker. Murad received his master's degree in life science at National Taiwan University and his PhD in molecular biology from Humboldt University, Germany in 2022. For his work elucidating, so he was working on elucidating magnesium regulation in the kidney. And in 2022, he joined Single Run Biotechnologies as a product manager. So welcome, Murad. And yeah, stage is yours.
Thank you very much for the kind introduction. As Dr. Ayman mentioned, today's agenda will be starting with the introduction of single-cell sequencing technologies. Why do we need single-cell sequencing at the first place? What are the application fields? And I will mention some techniques developed over time. And I will mention the workflow for single-cell RNA sequencing. So let's start with the first one. So I like this analogy. On the left side, you're looking at a delicious smoothie. And smoothie is made of different fruits. And on the right side, these are the fruits that makes up this the smoothie. So if you consider smoothie as a bulk RNA sequencing, each fruit would be single-cell RNA sequencing. So each cell-specific types would make up the smoothie. So with the whole smoothie, you get the taste, but you don't know the content of it. So in order to understand this, we need single-cell sequencing, basically. Single-cell was selected as a breakthrough technology by Science in 2018, and it was in the cover of Nature Methods for 2019. So it's breakthrough technology that help us understanding different heterogeneity of the tissues and different experiments. It opened a whole different era for us. So if you look at the publications on the PubMed by using single-cell and RNA sequencing as a query, you can see from 2018 to 2021, we see a dramatic increase in the number of publications. In the beginning, it started with 2, and now we have more than 2,300 publications published by using single-cell RNA sequencing. And as you can see here, the real development comes after 2018. So this is the part where the high throughput sequencing was discovered, which I will mention this in the later slides. So before I get into that, there are many different sequencing, single-cell sequencing methods available. Depends on your question, research question. If you're interested in epitopes that are on the cell surface, then single-cell CytSeq can be one option. If you're interested in histone chromatins, then single-cell ATAC-seq can be an option, or single-cell chip sequencing can be an option. And there are also different sequencing methods developed over time for different purposes. So it really depends on your research question. But the majority, if not more than 80% of the single-cell methods are focused on mRNA-based methods, on the transcriptome, detection of the whole transcriptome. And today I want to focus on those techniques that I mentioned here. So what are the application fields of single-cell sequencing methods? With single-cell sequencing, we can understand the heterogeneity of cancer cells and the tumor microenvironment. You know, cancer is really complex, and it consists of a lot of different cells interacting with each other and with its microenvironment. By using single-cell, we can decipher these interactions. So we can also use a single cell for neuroscience. We can classify the neurons. We can get information about cellular interactions, and we can use these information to understand neurological disorders. The other important application field of single-cell RNA sequencing is immunology. And for example, everyone, let's say 2 people get the same diseases, they get the same treatment. But one of them responds, the other doesn't respond. And the reason being is that each individual is different and everyone has its own immune profile. And today it is possible to understand this on patient-by-patient basis by single-cell RNA sequencing methods. So by using that, we can accurately identify immune cell subtypes, and we can reveal the immunological mechanisms of diseases. Another popular field that's where single cell is applied is developmental biology. People are interested in development of certain cell lineages. And to do that, we perform single-cell RNA sequencing, and there are more techniques that I can share with you today, or in the later webinars that we can cover those, that help us understand the developmental processes. So as I mentioned earlier, every human being is different from each other. And there, even within the same human being, there is a big intratumor variation. So in the tumor tissue, you can see that it consists of many different tissues. On the right side, you can see in more detail that there are many different cell types in tumor process. And these cells are interacting with itself, with the other cells, and with the environments. And with single cell sequencing, we can decipher those interactions. So what type of information can we get out by using single cell sequencing? One and most frequently reason to use single cell RNA sequencing is to understand the heterogeneity of the tissue. So if you check any single cell RNA sequencing paper, this is the graph that you come across on the right side. And this is t-SNE plot. Normally, single cell sequencing data is a multi-dimensional data. And here, what we do is we reduce these dimensions into 2, namely t-SNE1 and t-SNE2. So only based on 2, we classify the cells based on their similarity. So here, that means these cell types in the same cluster are similar and different than the other ones. Here you can see this is bone marrow cells, and they are classified, clustered until 20 different cell types. Another information that people get is how sulfate is determined. Especially this question is asked by developmental biologists because they are very much interested to know the cell development, cell lineage development. And there are different algorithms developed for this purpose. One is trajectory analysis, the other is RNA velocity. But based on those, we can identify progenitor cells, precursor cells, immature and mature cells, and it's depicted based on component 1 and component 2. The other question that people are interested in, which cells are more actively transcribed, which is similar to sulfate determination. And in this case, there is a computational approach called RNA velocity that can decipher this question. But RNA velocity does it, it looks at the ratio of unsplice to splice variants and say, okay, this transcript is too active, it doesn't have time to go splicing. So that means if it's unspliced, that means this transcript is too active. And we can get this information by single-cell RNA sequencing data. Another question that people are interested in, which cells are communicating with which cell types? Here on the right side, you can see this is a typical graph that we get out of this. There is a certain algorithms that help us to understand this question, but at the end, we get this type of graph. Here, that means the red color represents the interaction or the communication. And here, for this example specifically, macrophages and endothelial cells are interacting with each other compared to the other cell types. So this information can be useful for some people. So I want to share some applications, example applications here. Single-cell RNA sequencing was used used on sample biopsies obtained from 42 different patients with advanced non-small cell lung cancer. And here it is identified, so the researchers identified 11 major cell types. So they collect, this is a very nice landscape experiment that help us to understand the structure of the tumor. Another application is coming from developmental cell published in last week's And here, people looked at human fallopian tubes in the normal case, in the disease states, and they sequenced roughly 60,000 cells and identified 12 major cell types. As I mentioned earlier, we can do much more than that. So if you want, you can subcluster those cells to look at how they respond to certain disease. And this is what they did here. So they classified one cluster among these 12, and which resulted in 10 different cell types. And between disease state and normal state, so these sub cells were reacted differently. And they depicted this in different, by using different experiments, such as RNA velocity, as I mentioned earlier, and some more. So let's continue with the technologies. As I mentioned earlier, currently there is more than 120 different single cell sequencing methods, and there is no only one way to classify those. And here are just some of them that I can think of. So we can categorize them based on their throughput, capturing methods, and the length of transcripts. So for the throughput, as you know, it can be high throughput or low throughput method. Also, there are some moderate or middle throughput as well. But yeah, this is also present. For the capturing methods, it depends. So in the beginning, we were using only manual approaches like microscope and manually picking the cells. And later fax came and it automated a little bit for that. And now the era after 2018 is the microfluidics era, which enables high throughput. And based on length of transcripts, we can look at full length, 5' capture and 3' capture. Each has its own advantage and disadvantages that I will mention in the later presentation today. So let's look at the scaling of single cell sequencing, particularly for RNA sequencing data. So it began with the manual. So there was only one cell. In 2009, Tang and his colleagues investigated one single blastomere, which is picked by pipette manually. And that was a proof of concept experiment. Later on, different methods came up like AST-RT-seq, which enables us to multiplex our samples. Later on, some companies introduced integrated fluid circuits, which each cell is processed in one circuit. It was, compared to other two methods, it was more high throughput, but still considered as low throughput. After that, there are some robotics came out, and basically they use the same principle, but with less human interruption. So these four is considered low throughput techniques. And after 2015, there is nanodroplets. Then there are different techniques as mentioned here, drop-seq, in-drop, also commercial company 10x Genomics commercialized this technique. And this enables us to work with thousands of genes in parallel. And then there is microvell-based systems. We as a single run, we use microvell system that I will mention in the later presentation today, which is also high throughput system. So if you go again one by one, so this is the proof of concept experiment. As I said, they use only one single cell from mouse blastomere was processed. And here on the left side, you're looking at the flow or workflow. We start with one single cell, which is lysed, and then we use poly A, poly D capturing together with the known sequence. And after that, we remove this primer and add poly A. After addition of poly A, it enables the second strand synthesis with another known sequence, which will be used later for addition adapters. And after cDNA amplification, cDNA is shared or fragmented. And the main reason why we need to fragment our cDNA is that currently, majority of the systems are using Illumina-based sequencers, and Illumina is shotgun sequencing. So we have to fragment our DNAs in order to sequence them. And this is the step that we do. After cDNA fragmentation, there is an adapter ligation process. For this experiment in 2009, they used solid P1 and P2 adapters, and they sequenced this. They used full-length cDNA amplification for this process. It was only one, and so it was a proof of concept experiment, basically. So now comes the multiplexing option. Here, there is one single cell per well or per tube, and it is introduced in 2011, just 2 years after that. And here, this is the first study that introduces barcode. So with barcoding, we are able to multiplex the sample, and this is how they did it. And to my knowledge, in this article, they look at 85 different cells. In this workflow, they introduced template switching oligo, which was used for multiplexing the sample. And then they use single primary amplification to amplify cDNAs, and then they add Illumina P1 and P2 adapters for sequencing. And they sequence from only one site, from P1 site. And this is, for example, in the beginning, I categorized into 3 based on their length of DNA, length of cDNA, and that was a 5' capture, example for 5' capture. Now I want to introduce you one of the most frequently used system back in 2013. It's called SmartSeq. SMART stands for switch mechanism at the 5' end of RNA templates. And on the left side, you're looking at the workflow for SmartSeq. So initially, SmartSeq came out, I think, 2013 or 11, and then 2-3 years later, that was improved version of SmartSeq, SmartSeq 2. In this method, we start with the single cell is lysed, and then by using poly-T oligos, we capture the poly-A mRNA. And after that, normally for the reverse transcription, they use this MML virus, reverse transcriptase, which adds additional bases at the end. These bases are serving as a template for the second strand synthesis of the DNA. And this is what they did here. And they amplify this. After amplification, as I said earlier, due to the limitations of Illumina sequencers, we have to fragment our DNA. But here they use another enzyme called TN5 transposase. What it does is it's called tagmentation. So it tags and it fragments at the same time. Basically, after it fragments, it adds these tags, which are indexes and adapters. And later on, we got sequencing ready libraries. So what are the advantages of SmartSeq methods? Basically, it's a full-length cDNA amplification, and it allows detection of alternative splice transcripts and SNPs. And disadvantages, since there is no barcoding, we cannot really multiplex this system unless you need to modify. And there's a certain bias on transcript length because based on the PCR, amplification of over 4 kb is not as good as the short transcripts. It creates some sort of bias. And also, due to the PCR bias again, it preferentially amplifies high abundance transcripts. So then comes integrated fluidic chips. And this is lab on a chip system on a low throughput manner. And here one chamber contains one cell. And each individual single cells in the reaction chambers undergo the same process I described earlier. They undergo lysis, reverse transcription, and cDNA amplification. So it is basically based on SmartSeq method. And then subsequently library preparation has been done. After introducing integrated fluid circuits, people were interested in automating this. But again, this automation was based on SmartSeq, and they use fact sorted cells to start with. And it is possible to scale up a little bit by using these automated systems. Now we mention about the high throughput systems. So the first one is droplet-based systems. So in 2015, these studies came out, which describes high throughput droplet-based systems. And here what you're looking at is this is a microfluidic system. So here you see there are 2 different phases. This one is aqueous phase, and this is oil phase. So cells and reagents are pumped from one side and beads pumped from the other side or injected from another side. What happens is that this is a pressurized system. So once water meets the oil, due to the pressure, it creates this pinch. And then it compartmentalizes cells together with the beads. By the nature, in ideal scenario, what you want to have is 1 cell per bead. But most of the case, you will have only 1 bead, but no cells. And sometimes you will have 2 cells per bead, which is, this is called doublets, and you don't want to have this in your experiments. So later on, these compartments are, so here cells are lysed, reverse transcription occurs, cDNA amplification, again, as I described earlier, performed by using TSO, template switching oligo mechanism, and then library preparation. So what are the advantages of high throughput and low throughput if you look at the comparison basis? On the high throughput, we can investigate tens of thousands of cells in parallel. And it is compared to low throughput, the cost is lower per cell. It's important to mention. Cost per cell is lower. The disadvantage of high throughput is it provides only limited coverage. We can only get some information from that. But for low throughput systems, because we have only, let's say, we are limited to the number of plates, number of wells in the plates, like 96 well plate or 384 well plate. So we have a really limited number of cells. And those cells can be sequenced more deeper. So you can get more information from those cells. And by that, we can detect alternative splice forms and SNPs. But the disadvantage is that we are limited to a certain amount of cells. And the cost per cell is quite high compared to high throughputs. So it comes down to research question, what you want to investigate here while choosing the methods. So the summary of droplet-based systems, they are, as I said, they are high throughput. One can investigate tens of thousands of cells in parallel, and it's lower cost, but there is a certain limitations to that. It is not suitable for large cells or irregular cells. So if you're working with, for example, fat cells, neurons, or cardiomyocytes, or different cell types that are slightly bigger, that's a limitation because the diameter in that fluidic system that I described earlier is around 40 microns. So anything over that can be problematic. And if you're dealing with the cells with higher lipid content, that could interfere with this oiling phase. So this is also important for some researchers. So this system, this microfluidic system is pressurized. It's based on the pressure. And for certain cell types, this could be a problem. It could damage those cells, and especially, for example, neutrophils. And the droplet formation may not be too suitable. And also in the literature, some researchers reported that it is likely that clogging would occur in the droplet-based systems. And finally, we come to the microwell plates. Here we, Singularn as a company, we use microwell-based system. And our technology is centered around specially designed barcoding beads and the scope chip here. So here, this is the real-life image of our microchip. Here you can see this is the size of a microscope slide to give you an impression. So it's big as a microscope slide. And here, white part you see here consists of thousands of wells depicted here. And you can see there are two, one inlet and one outlet. So you can inject your cells from here. It flows through. And during this flow, cells, thanks to gravity, they sink. So this is the major difference, one of the major differences between droplet-based system and microwell-based system. In a microwell system, it is gentle to cells. So you don't pressurize anything. And here, this is a typical bead structure. It contains PCR primers for addition for PCR, later step addition of adapters. It contains cell barcode. So cell barcode is necessary to identify the origin of the cell, because once they are broken, each cell should contain only one cell barcode. So UMI stands for unique molecular identifier, and it's 12 base, and it is given to every RNA molecule in the cell. So one molecule will get one UMI, but different. So in each well, it will be multiple UMIs, millions of different UMIs, but only one cell barcode. So this is how we identify or trace back to the cell type. And then it contains poly-T for capturing polyadenylated mRNA. After the capture, cell lysis and barcoding occurs, followed by reverse transcription, amplification, and library construction. I will mention this in more detail later on. And here is a real-life image and how beads are looking under the microscope. So for the empty beads, you can see that. And once you load the cells, cells are following this Poisson distribution. So number of wells are way too much compared to the cells that we load, so that we minimize the risk of having two cells in one well. You can see cells are really separated from each other. And then we code the wells with the beats. For the beats, it's not important. Based on the engineering, each well can contain only one beat. That's pure math. So the length, so the width of our well is 40 microns, and for the beats are 30. So only one beat can fit in. So now let's look at the single cell sequencing workflow. So this is a general workflow, as I mentioned earlier. You start with the tissue, you extract single cells, suspension, and then we load this into our handheld device, microchip. And this is where we partition our cells, where we distributed cells to the wells. And cell lysis and mRNA capture takes place here. And then we perform reverse transcription, cDNA amplification, and library construction. And at the end, it is ready for sequencing and bioinformatic analysis. So our kits cover from tissue to sequencing ready library preparation. So let's walk through each step one by one. So if there is one step that is important in this workflow, is the tissue preparation step, or single cell suspension preparation step. And it determines the outcome of the RNA sequencing experiment directly. So if there is one slide that I want you to remember from today's presentation, it will be this one. So as you know, there are different tissues, and each tissue needs to be optimized specifically. But for this purpose, we develop S-Salive reagents that contains tissue preservation and tissue dissociation, which suits the universal needs. For people who are interested in automation, we have also an instrument for dissociating tissues, and that can perform the entire workflow under 15 minutes. So the most important thing when it comes to tissue dissociation is to get high cell viability. So you want to aim for no more than 90% if possible. And it shouldn't be lower than 80% based on our experience. So with Python instrument, we can process 8 samples in parallel. So to give you a little bit more detailed information about tissue preservation and dissociation reagents, so here we have developed this nice tissue preservation buffer, which can effectively preserve tissue for 72 hours at 4 degrees. So if you're collecting samples on multiple days, what usually is the bottleneck for RNA sequencing tissue preparation is once you take out the tissue, it starts differentiating and it starts expressing certain genes. So the transcriptome is affected, and you don't want to have this effect. So that's why almost all of them, unless you're going for frozen tissues or nuclei, that requires you to have fresh samples immediately processed. So we want to solve this issue by coming up with this tissue preservation buffer. With that, you can keep your cells, keep your tissues for 72 hours at 4 degrees, and it contains no chemical fixatives. And here you can see the data. In fresh, 90% is the cell viability in the beginning. And now after 72 hours, it only drops 2%, so 88%. And on the right side, this is the UMAP or t-SNP plot that I described earlier. And you can see in 24 hours, 72 hours of fresh samples don't have any difference in between. The samples or T cells are overlapped with each other. For tissue dissociation master mix, we come up with this master mix that works with more than 300 different tissue types, and it can effectively dissociate these cells under 1 hour. And you can see after 1 hour, the cell viability is still significantly high compared to other commercials and the techniques that are reported on the literature. So the second step is single cell suspension. And here, as I said, if you get your cells from the tissue as the methods that I described earlier, it shouldn't be a problem. Then the next step is partitioning those cells into these microfluidic chip. So for that, we have different options. We have standard chip, which contains 150,000 wells that can capture up to 10,000 cells. If it's not enough for you, there is also high density option, which contains 250,000 wells that can capture up to 30,000 cells. And they are in 40 micron in size. But if you're dealing with the different cell types that I described earlier, we also have an third option, which we have larger wells that you can work freely with. And again, this is the same image I described earlier. The next step, so for this step, cell partitioning, you don't need any kind of special instruments. All you need is a P200 pipette, which every lab has it. For people who are fan of automation or who are processing a lot of samples at the same time, there is an automation option as well. So we developed the single row matrix instrument for processing 2 chips at the same time under 40 minutes. The next step is reverse transcription, cDNA amplification, and library construction. So for this, I will mention this in more detail, a little bit more detail. So here, this is the bead structure. It contains, again, poly-DT, UMI, which stands for unique molecular identifier, which is help us to describe each transcript, and the cell barcode. For the cell barcode, as I said, each cell contains the same cell barcode, and for the read, for the later steps. So we capture polyadenylated mRNA, and then reverse transcription occurs. At the end of the reverse transcription, by using this MML virus, it adds certain, like 3 or 4 nucleotides at the end. And we provide template switching oligo as a template for synthesizing the second strand of cDNA. After the reverse transcription and cDNA synthesis, we amplify the cDNA by using 2 different primers, one on the read one, the other on TSO. So after amplification, as I said, we need to shear the cDNA or fragment cDNA. After fragmentation, end repair and A-tailing occurs. And we ligate the adapters for sequencing P1 and P7. And at the end, we have Illumina sequencing ready libraries. So for the sequencing part, as I said, currently, majority of the markets, almost all of them, are using Illumina systems. And currently, there's a certain limitations to that. The latest technology from Illumina, the NovaSeq, it can only does 250 base pair in pair end mode. There are some long read sequencing options that I haven't mentioned here. I didn't mention it because at the moment, technology is not there yet, quite there yet. The error rate for sequencing is very high. And if you're dealing with the cell, like individual cells, like thousands of cells at the same time, this error rate like multiplies, and you don't want to have this. It's basically highly erroneous. So that's why we are still limited to short read sequencing. So after you sequence your samples, you get your FASTQ files that are, in this case, we provide software that is deposited freely on the GitHub called Telescope, which help you to visualize your data, query the database. And we also developed the Synecosis database, which help in separation of the clinical data, or it helps you to classify or cluster your data. So what are the advantages of microvel-based technology? Microvel is high throughput, it's same as droplet base. And with our technology, we can capture up to 30,000 cells. And it is ideal if you want to discover some rare cells, if you want to work on the rare cell populations. It's a manual method. You don't need any special instrument, but if you are up for it, there is an possibility for having automated manner. It is versatile, so you can apply this to different cell types, different size, different cell content. And the thing that I like most is like QC during the workflow. So let's say you partition your cells and you see that cell loading wasn't good, so you can reload the cells, or you did the beads and it wasn't enough, so you can do it again. All you need to do is just take your microchip and go to a microscope and look at under the microscope. It is that simple. You don't need complex instruments to scan it or to visualize it. It is flexible, as I mentioned. The size of the microvals can be changed in comparison to the size of microfluid channels in other systems which are fixed. And it is open system, so it allows you to modify the techniques based on your needs. So you can introduce various reagents during the workflow to test something out. You can just wash them out at the end. So it gives you this flexibility. And so far I talked about the only RNA sequencing part, but as a company, we have more than that. We offer more than that. So I talk about the single cell RNA sequencing part. And we also offer single cell nucleus RNA sequencing part. And we offer single cell immune repertoire profiling. As I mentioned earlier, for precision medicine, in the immunology, it is very important to understand the immune profiling, immune repertoire. And with this kit, we are able to do that. So we have these two unique products, namely Focuscope and Dynoscope. So Focuscope is for targeted sequencing, and it not only detects the mutations, it detects it with the 3' RNA sequencing. So it detects the whole transcriptome together with the mutation. Or same thing goes for the fusion genes. And you know, fusion genes are important for, especially for certain tumors, certain cancers. And our products is, I think it's the only on the market that can detect fusion genes together with the whole transcriptome. So we are not only limited to poly-A mRNA, we can also detect some viral sequences by that. This is a targeted approach. So another unique product is called Dynoscope. Here we add time to the transcription data. As I mentioned earlier, there are computational methods which can do the similar thing, like RNA velocity, for example, which determines the time of the transcript. But here there is a chemical approach for time-resolved transcription dynamics. And that's all from my side. Thank you very much for your listening and your participations.
Yeah, thank you, Murat, for the comprehensive presentation. So I would like to encourage everyone to be interactive and ask any question if you have. So let's start with the questions. We have some questions here. The first one is how to choose the right technique for the experiment.
How do we choose the right technique? It all comes down to research question that you're interested in. For example, in the beginning, I introduced you high throughput and low throughput systems. And the main difference is the gene coverage that they offer. So let's say the cell type that you're interested in, if it's expressed, if it's not a rare cell type, you don't need to sequence too many cells. And in this case, you can get away with low throughput systems. Also, it depends on what type of machines or what type of technology you're interested in. And for some techniques, and most of the techniques, you need to purchase bulky machines, instruments to perform those. Also, this is something that one needs to consider. And that's the things that I can think of at the moment.
Yeah, so how to make sure that one cell or bead present in each well?
So as I described in my presentation, our technology is based on this microvell system that contains hundreds of thousands of wells. And comparing to the number of cells that we load is very low compared to the number of wells that we have. That assures you that it follows Poisson distribution. So at the end, majority of cells beads, so majority of the wells will be empty. Majority of the other, let's say, the other part will contain only one cells. And when it comes to beads, this is the width of our well is 40 micron, and each bead is 40 micron. So that assures you to that is making sure that one bead can fall into one well.
What is the necessary sequencing depth?
So the sequencing depth is also depends on the research question. And usually, if you're going for high throughput systems, between 20,000 reads per cell to 100,000 reads per cell would be sufficient. but majority of the cases on the literature, it's around 50,000 reads per cell. On the other hand, if you're going for low throughput methods, then you want to sequence it more deeply, and this is where the more information can come. So if you want to sequence, if you want to choose low throughput methods, then I would recommend around 1 million per cell.
Yeah, and what is the difference between in drop system and dropseq?
So droplet-based methods, there are different methods in dropseq or dropseq, and the main system among them are the same. The main workflow almost the same, but they differ in the material that they use. So they use one for dropseq, they use this brittle resin, and for in-drop, let me think, there's another difference between, it's about the primers that they use. One is photocleavable, the other is tethered to the cell surface. So there are some technical details there. But the things that most of the users care about is the number of genes that they can detect. And for dropseq, it's usually superior to in dropseq. One can detect around 2,500 cells. And for the in dropseq, one can detect around 1,200 or 1,300 cells. And when it comes to workflow, also there is some differences there. It's a dropseq workflow is relatively shortened than in dropseq, so that would be the changes in between.
If there is any more question? Yeah. So they also, so it is also asked for the video of the meeting or the slides. Yes, it will be uploaded to our YouTube channel. So you're welcome to subscribe to our channel. Thank you again. And you're welcome to contact us if you need any consultation or any question. And also you can also visit our website or follow us on our social media. Thank you again for joining us. Have a great day.